I want to go very quickly over what we will say at this point about tannins and a little bit about lignins, and then spend the rest of the period on the chemotaxonomic handout. Tannins, of course, can be divided into two subgroups that we've already discussed: the hydrolyzable, in which the linkages of the phenolic acids are by ester linkages to hydroxyl compounds. The hydroxyl compounds are most commonly glucose, but may be quinic acid, carbohydrate-like alcohols, and so forth. So that, for the most part, the hydrolyzable tannins are derivatives of gallic acid, at least the ones we know best. And it's intriguing that hydrolyzable tannins, except for epicatechin gallate, which you might call a hydrolyzable tannin, at least has a unit, something like a hydrolyzable tannin. Except for that, they don't occur in wine, and yet tannic acid is the only thing that's legal to add to wine, which has a historical basis we may mention some other time, but it seems kind of ridiculous in a way. The tannic acid that you buy is considered to be Chinese tannin, according to your notes, at least that would fall in this group, and would have around 9 gallic acid units per unit of glucose. If glucose was completely digallic acid esterified, then you'd have 10 gallic acid units per glucose unit. The biggest single unit that's found in the mixture, and it is a mixture, is generally about 7 gallic acid units on 1 glucose. So you see, and it is a heterogeneous mixture. On the other hand, you do find, according to the notes here, about 7% metadigallic acid, which of course would raise the gallic acid percentage. So if you just analyze the mixture for sugar and for gallic acid, you get around 9 up to 10 gallic acid units per unit of glucose. That has some significance in terms of analytical use of gallic acid and so forth, but it just illustrates the heterogeneous nature of the material. Another point of interest is that the Chinese tannin is caused by from a gall caused on a Rus genus plant, and you remember the Rus is a genus of Anacardiaceae, like Rus toxicodendron, which is the poison ivy, and this is a similar plant. I'm not so sure whether it has any skin irritation effect or not. And this galling effect by a specific aphid on the leaves, then the galls are separated, and gallic acid gets its name from coming from galls. And many galls on many plants, are very high in tannin. And this is something I think needs to be studied. To my knowledge, it hasn't been, in that the insect lays its egg, causes the gall then is very high in tannin, and that seems to protect the insect that lives in the gall from other insects that tannin would be obnoxious to. So that there is a nice, from the insect's viewpoint anyway, symbiotic or quasi-parasitic arrangement going on between the two. Well, yes?
Is the manufacture of this tannin an industry by itself, or is it a byproduct from,
No, it has been an industry by itself. They collect the galls and isolate the tannin from the galls, and it's been used for hundreds, if not thousands of years, to make ink and tan leather and so on. So that it is still largely a cottage industry type thing in terms of collecting the galls and then bought by some central purchaser and then processed later on. Of course, it's not a big item in commerce any longer, because most of the leather is made by non-vegetable tanning rather than vegetable tanning. And in the same group we have then sumac tannin, and notice again sumac, if you grew up in the Midwest, you know what sumac is. It's a small bush that grows along the roadside with a nice seed head in the wintertime, rather pretty red decorative seed head, and again a Rus species, a member of the Anacardiaceae. Basically the same chemistry as the gallotannin from Chinese tannin. I'm not quite sure what the situation is, since Chinese tannin is correctly named. It originally came from China, and with all the embargoes and difficulties in commerce with China, how much of it is still Chinese tannin, and from that source I'm not sure. I think a great deal of the tannin that we get as tannic acid recently has been the so-called Turkish tannin or Aleppo tannin, and it's a similar situation, except it's a different insect. I believe Cynips is a kind of a wasp. I'm not certain, but in any case, this insect stings the twigs, and then characteristic galls develop on an oak tree. And the oak tree is called Quercus infectoria, the infectoria coming from the fact that these trees have so many galls on them that they look like they're bearing fruit. And you may have noticed in the valley oaks in California, you find some trees, and most trees have a few. Some trees in the foothills will be so heavily infected with galls or infested with galls that they do look like they're bearing fruit other than acorns in the wintertime. Doesn't seem to hurt the tree much. What the insect is that infects ours, I'm not sure. If we ever get short of Turkish tannin or Chinese tannin, I suppose we can get tannin from our own oaks if we wanted to go to the trouble of gathering all the galls. Terratannin is not the result of a infectation and a gall production, but rather is directly from the leaves, as for that matter is sumac tannin, so that the tannins are present in the plant even if there is no galling effect, but ordinarily the galls are much richer in percent by weight. The galls run up to 30% or so of the dry weight as tannin, whereas the leaves and other parts are seldom that high, more often around 10% in the high tannin materials, and then of course a good deal less in the plants that are not used as sources of tannin. The breakdown type products like glucose monogallic acid and so on are found in certain other plants, and often as partial hydrolytic products from the tannic acids. Since they are hydrolyzable, they are rather easily broken down, and even in the commercial product there is some of the partial hydrolysates. The next group in the hydrolyzable tannins would be the enology tannins, and as the name implies, they would break down to give some ellagic acid upon hydrolysis. And examples of several myrobalans, which is a kind of a plum-like fruit from a semi-tropical tree, seed pods called divi, which is a legume plant, algarobilla or bia, which is a South American tree. It's a acacia relative. I'm not so sure that isn't the one that's called kiavi in Hawaii. I think maybe it is. If you've been to Makaha Beach for the surfing contests and were barefooted as you walked along the beach, you know what kiavi is, because it's pretty thorny, and it has seed pods that are certainly very tannic, and I believe it may be the same one, and in any case it's a similar tree. Chestnut bark has been used for a long time, and wood to some degree, as a leather tanning agent, so that the trees were cut down, bark stripped, and even the heartwood ground up. And then you've heard of tan oak. Tan oak is similarly prepared. There is a tan oak that grows on the west coast, which is a different species than Quercus. It's a lithocarpus, but it's related to these oaks. And these were used as sources of tannins in vegetable tanning of leather, still used to some degree for sole leather and very high quality thick leathers. You may have heard of the tan bark when the horses or whatever are in the show ring. Well, they had so much bark left after the extraction of the tannin that they used the tan bark then for making dust-free show rings for horses, etc. So you still see in the sports pages, etc., people talking about the tan bark. Well, that's how the term got going. Still find a few. I remember the State Fair in Indiana still did use tan bark the last I knew, but I don't know where they get it because it's a lot less common. That's a fortuitous sidelight that no extra charge for that. The correlation is the first specific structure given for an ellagy tannin, and you'll notice that the Ellagic acid is not present as such in correlation. You'd have to hydrolyze the diphenic derivative of the two gallic acid units. Notice the two gallic acid units that are at the lower part of the molecule are cross-linked through the two ortho positions. Then if you hydrolyze this from the sugar, you'll get the diphenic acid unit, and then that spontaneously lactonizes in solution to ellagic acid, and therefore it is an ellagic tannin. That's generally true. Ellagic acid itself is not possible to esterify with sugars, so ordinarily the ellagic tannins are gallic derivatives, but derivatives that have already been cross-linked to make the double unit that then becomes ellagic acid upon hydrolysis. That's not invariably true. If you look on page 47, valinaic acid dialactone is mentioned. Well, that is an ellagic tannin that has a ether-linked gallic acid to an ellagic acid. However, it is a breakdown constituent, or an artifact, if you like, from hydrolysis of valinea tannin, which is an acorn cup tannin. The optical isomers of the diphenic acid, as mentioned in item 9 on page 47, are known, and the reason there are optical isomers is you have restricted rotation about the linkage. So if you have the two rings and then you have the carboxyl group and the OH groups, they can't rotate past each other. Seem to be short of chalk again. Now here it is. So that we have the diphenic acid type linkage, and you have a carboxyl group here and an OH group here, and that's why it lactonizes so well. And an OH group here. So the molecule cannot be in the plane of the blackboard as I've drawn it. One ring has to be at right angles to the other ring, and then depending on which way, which of the two possibilities it has formed in, then it will be optical isomers. And naturally, they do tend to occur as one or the other, which shows again that the ellagic acid is present in the mixture. Of course, ellagic acid itself cannot have optical isomers, because by forming the lactone, we would complete the ring, and then the molecule has to be planar. Ellagic acid itself is very insoluble and has been shown to be a cause of a turbidity in certain wines, notably Loganberry wine, and the fact that ellagic itself is so insoluble would mean that it wouldn't stay in solution and get into the bottle and cause trouble. But the ellagic tannin can stay. It's glucose-bearing and rather soluble, and even the diphenic acid may be water-soluble enough to stay in the solution long enough to get in the bottle. Then the lactonization, or the hydrolysis of the tannin followed by lactonization, gives an insoluble product. And turbidity in wine that can be of considerable magnitude. I could bring in a bottle of Manischewitz Loganberry wine that has about a teaspoonful of white deposit in the bottom that is ellagic acid, and it makes the wine very unattractive and questionable.
Is there any way to find that out?
It's easy to filter out once it's formed, so the trick is to get it to form completely, or fine it out by some gelatin type fining early, or better yet, prevent it getting in there. Apparently the tannins are confined mostly to the seeds of such fruits, and the seeds are dispersed in a Loganberry like in a blackberry or whatever, and so the idea is to press the juice off the pumice, don't ferment on the pumice. And such tricks as this, it can be and has been solved apparently as a production problem. I worked on this and published a paper, and I got into more trouble with that paper than anyone I ever did work on, because on the one hand, the commercial winery, Manischewitz, that we worked with, decided, they agreed in advance, but after we published it, then they decided that they wished we hadn't published it because it sort of suggested that there was a problem with their wine, and they didn't like it for that reason. And then the grape wine people didn't like it because they said, well, here we're supporting your research for grape wine and you're off working on Loganberries. Where do you get off doing that? So I dropped it like a hot potato, and we haven't done anything more with it. Otherwise, I'd be able to answer that question a good deal better. An example of where some easy research looks like fun, and then people take the fun out of it by quibbling about details, it seems. Anyway, the condensed tannins then are a quite separate group, and they're all polymers of other than ester-type linkages, and these other linkage are generally, as far as we know anyway, carbon to carbon and sometimes ether linkage that are not easily hydrolyzed or completely non-hydrolyzable. In fact, if you try to hydrolyze this kind of a molecule, instead of getting monomeric materials, you'll get further polymerization. So if you heat a condensed tannin in an acid solution or an alkaline solution, worse yet, which would be the way of hydrolyzing a gallotannin, you'd end up with what are called phlobophene, which are nondescript, completely insoluble phenolic humic acid-like materials that get the name phlobophene because they also occur in bark. Phlobos is a Greek word for bark. And if you grind up bark and try to extract the brown tannoid materials from it, you'll find that they're pretty insoluble and pretty inert to most things. They're not lignin, but they're tannin-like, and yet they're big and insoluble, and this sort of thing results from oxidative and attempts to hydrolyze to give degradation in the condensed tannins, not so much in the hydrolyzable tannins. These are a more heterogeneous group. There are several examples from acacia, this acacia catechu, to which the name catechin comes from. I suppose it ought to be catechin because I think it is called acacia catechu, but in any case, there's where the name comes from. It was first isolated and is still commonly produced from gum catechu, which comes from the acacia family, acacia genus. And several other trees, black and such things as black tea tannin can be considered to be a condensed tannin. How much is artifact and how much is in the tea plant is subject to argument. And many of these give, most of them, if not nearly all of them, give leucoanthocyanin reactions. So if you heat these condensed tannins in an acid solution, you'll convert them partially to anthocyanidins and therefore a red color. So that if you want to test for this kind of thing, that's one quick test. A little bit of the tannin material in an acid solution and heat it. If you get no red color, its chances are it's a hydrolyzable tannin. Of course, you could go further and look for gallic acid in the products and so on. There are a few strange members of the group on page 48 under uncertain or mixed condensed tannins. There are apparently still bean derived tannins. In Picea abies, which I believe is white spruce, anyway, a spruce, about 16% of the bark is a tannin material which appears to be made from still beans rather than from flavonoids. But that's about the only case that I know about. The rest of the tannins are, except for the ones that come from steel bean and their limited distribution, they are all derivatives of either catechin or leucoanthocyanins in polymeric arrangement. They may include, at least when you isolate a group of polymeric phenols, you include in that some possibly extraneous units. For instance, a small amount of glucose is generally found, and whether it's a glucoside on some of the flavonoids or whether it's an extraneous artifact is a little bit hard to determine in many cases. This quebracho tannin is kind of interesting. It's one of the most widely used of the vegetable tannins that are still in use. Quebracho, as I understand it, is Spanish or maybe a Latin, slang Spanish. It means axe breaker. The tree is very hard, and this tannin comes from this Anacardiaceae again, and these trees are harvested for tannin from the wild condition in the South American countries mentioned in your notes. It contains a high percentage of catechin, but is also some leuco content, and it seems to contain some hydrolyzable components. So it's a completely mixed tannin, apparently, in the sense that it has both groups. Ordinarily, that doesn't happen, and it may be the way the material is isolated. It's quite common to find a hydrolyzable tannin, say, in the leaves, and a condensed tannin, say, in the heartwood, so that can occur. But more commonly, you find one kind in a given tree. Say, for instance, in the oak, that is oaks as used for barrels. Cheryl Markman has just been finishing some further work, and it seems quite clear, and our analytical work has shown that the tannins of oak heartwood are hydrolyzable tannins that already been shown, and of course these galls and so forth indicate that leaf and bark tannins are essentially completely hydrolyzable tannins. So this enables us then to analyze wines which contain no hydrolyzable tannins for their hydrolyzable tannin content, and then we can use that as a measure of barrel aging, for instance. So that, or it could be tannic acid addition, but barring tannic acid addition, it would be an indicator of barrel age or wood contact, chip treatment, that sort of thing. Now, the polymerization and the nature of these linkages, particularly, we didn't say much about hydrolyzable tannins beyond that they're ester groups, and that makes it fairly obvious. In the condensed tannins, there are a more heterogeneous group of linkages, and I'm going to come back to this later on when we talk about polymerization in wine and so forth, so I'm not going to say much about it at this point. Just to point out again that the linkages are primarily carbon to carbon with some ether type linkages, and there are several different kinds of linkages, but the three main ones that have been suggested are as on the top of page 49, with a link of one flavonoid to the open chalcone form in the 2 position of another flavonoid unit. Now that has been shown to occur, but it seems a good deal less likely than some of the other alternatives, and another one is shown under item C as a link between 2 B rings of 2 compounds. This certainly occurs in the bioflavonols, and there is evidence that you can produce this kind of product in the test tube, and Hathaway has some evidence that it may occur naturally. But these both are less likely, and we think generally in the true tannins of the kind we're finding in nature, that the linkage would be between the 4 position of one unit and the 8 or 6 position of another unit. So the most common natural link, maybe the exclusive natural, and these other things may be artifacts, would be this sort of a link, where either the 8 position or the 6 position are activated by the meta-OHs in the sense of being readily attacked by a positive. They are nucleophilic or have a high electron concentration, and the 4 position tends to be positive, and we can get polymerizations and perhaps depolymerization from this condition. And in the case of the Flavan 3,4-diols, this polymerization seems to be a dehydration, net dehydration type reaction, in the sense that the elements of water are lost, the OH from the 4 position and the hydrogen from the 8 or 6 position. And apparently this is why the Flavan 3,4-diols and leucoanthocyanins are in this kind of structure and form and degenerate from this structure, but a certain amount of catechin is present. But every time you have a catechin, you would terminate the chain, because having 2 hydrogens here, it's not capable of producing this reaction. So one of the things that one might do when you convert a polymeric leuco to anthocyanidin, you commonly also get some catechin. Unfortunately, you can get catechin from some of the other units by other type reactions, but one thing that I've always thought it would be well worth looking into, and to my knowledge nobody has yet, if we determine the ratio of catechin to anthocyanidin after leuco conversion, we ought to be able to work out some method of polymer length determination, because the polymer length would be it would be 1 catechin per unit of polymer. So far that hasn't been done, and there are good reasons why it hasn't by me or anybody else, and that is that the reaction is pretty messy, and you don't get a very high and very good yield of conversion to anthocyanidin. On the other hand, it's certainly something that would be very helpful, because by and large we don't know the molecular size of these things, and we have some interesting suggestive data that suggests from the grape mainly that the polymerization is not strictly random. In other words, we seem to get in grapes, we have dimers, and then we have something like maybe tetramers. And then there is some evidence for maybe octamers. I don't mean to say that they're going up in units of 2 necessarily, or doubling, but on the other hand, the evidence is you don't have everything. If it was strictly just random chemistry, you'd expect 1, 2, 3, 4, 5, 6 right on up the line. And it seems you don't have that, which means that there ought to be some enzymatic control of the system, and that brings us to another problem. How would you, if you were God and you were setting this thing up, how would you go about arranging to have a proteinaceous enzyme make a protein precipitate? You see, precipitant, it would be pretty tough. And so whether or not it's enzymatic control is a point of some importance. If it is, how can it be done? This is a little philosophy, I guess, rather than chemistry, because we haven't worked out how we can even determine this for sure in a very satisfactory method yet. Let's go now to lignins, and I want to spend even less time on lignins at this stage. We may come back to them later on. One reason for spending less time is that it's to a degree a dead end, and there aren't many applied importances to lignin except in making paper, where lignin's a big nuisance and you're trying to get rid of it. Biochemically and in the plant, it's a big mystery to some degree, and we'd certainly like to know more about it. First point to be made is that lignin is not a compound, so you'll never be able to isolate and crystallize or whatever lignin, because lignins are a group of apparently heterogeneous materials, a 3-dimensional polymeric unit, and the polymers are again mainly carbon-linked and or ether-linked, which means there's no simple way to break them apart. If you use drastic enough treatment to, quote, hydrolyze, unquote, a lignin, you would have broken it down so extensively that the artifacts outnumber the true fragments, and you don't really know where you are. The best studies on lignin have been the other way around, and largely conducted by Freudenberg in Germany, and his followers were co-workers and students, and Vigne is one of them, who's doing the best work currently, I think, and has worked on grape materials, grape dimers, and the tannin type, condensed tannin type of compounds. And the way they've done this is to take coniferal alcohol and or relatives, and it seems that the main tannins come from coniferal alcohol, and treat them with hydrogen peroxide and peroxidase. And apparently the only biosynthetic role of enzymes in the formation of lignin is to initiate the dehydrogenation, excuse me, the removal of hydrogen or the oxidation of the compounds by a peroxidase type enzyme, and the best evidence now it is peroxidase, and this is one of the main known functions, or fairly newly known functions, of peroxidase in plants, is that it is apparently the source of initiation of lignification. In any case, these compounds are oxidized, that is the coniferal alcohol type compound is oxidized, and a lignol type of quinone methide free radical structure apparently develops, as is illustrated on page 51. This quinone methide will link to give dimers. These are called dilignols, and then these can link further to make heterogeneous 3-dimensional polymers of the type that ultimately is indicated on the bottom of page 52, where we've tried to show in a fairly compact picture all the possible linkages that have been reported in lignin. And you see there are quite a great number of different kinds, different side chains, linking through the phenolic ring, linking through the 3-carbon side chain, and cross-linking in various different ways. And again, it's a 3-dimensional plastic-like material, which is very difficult to study. And the most satisfactory studies have, as I say, been going the other way, taking a coniferal alcohol solution and forming the initial lignin-like molecules and then studying those, because you can limit the possible reactions a little bit more. While we're on lignin, I should mention, and if you want to add to the understanding of what I'm going to say, the mention is under item B on page 50. The natural occurrence of lignin, however, has some significance. It is cinnamic acid derived, and there are mainly 3 kinds of lignin. The Gymnosperm Lignin, and the gymnosperms are the more primitive higher plants, if you like, including the conifers. cycads and plants of that kind. The gymnosperms generally have only the coniferal alcohol type phenolic structure. That's the guaiacol type benzene ring. Whereas hardwoods, angiosperms that are more evolved, have that, but also in addition have the Snik Acid derived lignins, and then the monocotyledonous grasses, bamboo, etc., have, in addition to those two, the paracumeric analogs. So there seems to be an evolutionary sequence making it the lignin more and more complex by adding to the kind of lignin that is produced in the more primitive plants. And we're talking now, of course, lignin is not present in the truly lower plants, so that lignification itself is a development of higher plants compared to lower plants. And then within the higher plants, the lignin gets more and more complex as the plant is more and more evolved. I think that's about all we'll take time to say about lignin, so let's turn then to, yes, question.
Did I miss something, or did you not say anything at all about natural grapeseed tannin?
I didn't say anything at all about natural grapeseed tannin. It's a condensed tannin, and at this stage we can't tell you much more about that. It's completely condensed. There's no there's a little bit of gallic acid again from epicatic and gallate inclusion apparently, but it's very small. We'll come back to that as we talk more about grapes and wine, I think. Okay? Just so you've heard of them, I thought there were a couple of units that I should add. The dibenzofurans are not very important. They were discovered in higher plants after I originally wrote up these notes. That's why they're not included. But they're C6 compounds. No, there's a bridge between the two rings, but it's an oxygen and a carbon bridge, not further carbon units. So they're an unusual group. They were known among lignins, and the one shown here is Snik Acid, which has been used as a medicine, sort of an antibiotic, from a lichen. I said lignin, lichens. This is from a lichen from Finland and other such places, reindeer moss so-called. And reindeer moss had been used for a long time by the Laplanders as a wound dressing and so on, and it turns out that at least one of the components is Snik Acid. No great significance, either medically or anything else other than a curiosity of some nature. The known example in higher plants is what's called the Australian finger cherry, and it's apparently not grown, certainly not grown as a fruit plant. It's a wild plant, the tree that grows in Australia, but they had a Australian Army what we would have called maneuvers in some outback area, and 20-some-odd soldiers ate the fruit from this kind of tree, and out of the 20, whatever it was, I think 23 of them turned completely blind and never recovered. They were permanently blinded, and it was from consumption of a derivative of a dibenzofuran. So in those cases, it was a very serious thing, and something that illustrates that you better not eat anything strange if you don't know what it is in the plant world, because drastic things can happen. More importantly, perhaps, although still very sketchy distribution, almost confined to the Dalbergia family, or genus, which is a very small tropical tree group of plants, are the neoflavines and the 4-phenylcoumarins. And you realize that if you look at the structure, this neoflavine is the further migration. We said isoflavones would be the migration of the phenyl ring from the 2 position to the 3. If the same sort of migration occurred and it went to the 4 position, then you'd have this kind of compound, and you could also have the 4-phenylcoumarins like Dalbergan. And these compounds have been found, as well as the chalcone analog, which would be this kind of compound here. Again, notice that they're C6, C3, C6 compounds, and they have been shown to be produced from phenylalanine, and apparently it is by the continuation of the isoflavone type shift. They're curiosities at this stage of very limited distribution, but quite an interesting addition to the family from a chemical structure viewpoint. They are somewhat important, and the reason they were studied is that this particular kind of wood is very resistant to decay and termites, and apparently it's because these flavonoids are so strange that the decay organisms and the termites can't metabolize them, and therefore helps protect the wood. Well, that doesn't leave me much time to turn to the evolutionary story. We'll probably have to add to our comments next Wednesday, but some of the things that I want to say relate a little bit to your pass-out test, so I want to spend a little time on it. You have, as usual, a list of some significant references, I hope significant, and if you want to look up anything further, this gives you some fairly recent reviews and books, as well as a couple of suggestions about continuing new paper series. And the Phytochem Journal has a section in it devoted to, I mean, you can look down the index and there is chemotaxonomy as a subhead, so you can pick out the work there very easily. An important point to be made is that phenols have been classified as secondary metabolites as opposed to primary, and I think it's important to see what we're referring to here. Primary metabolites are those that are necessary for the system to live, so it's essential for life, and the essential for life compounds would certainly include tyrosine, since it's an essential component of proteins as we know them. So there are phenols that would have to be called primary metabolites. But most of the phenols are not found in all plants, or even all plants of a certain kind, and therefore they must be considered secondary. If they aren't present in everything, they can't be essential for life. That being so, are they important or not? Well, the original idea, as I've mentioned before, was that no, they're not important. They're just kind of curiosities or jokes Mother Nature played on chemists, and you know, it's really not too much importance to them. They're sort of excretory products, detoxification mechanism, and the plant has some, you know, they don't do things very efficiently, so they shovel these things off into some cells and sort of forget about them. Well, that was all right if you didn't want to use your head any, but it seems to me that the more positive approach it makes a great deal more sense, and more sense as time is going on. And by the positive approach, I mean that they must have some function or they wouldn't have survived. Now, the function might be a detoxification or excretory type function, I suppose, but if that's so, why don't all plants then have such compounds? Because clearly if one plant needs to excrete something or shovel it off into an unused cell, why not all plants? And of course, lignin obviously has a function to help hold the trees up. The grapevine is low in cellulose, I think, instead of lignin, but the reason it must climb on other trees is that it isn't strong enough to stand up as itself. So you have to stake it or grow it on a tree, as it does in nature. Well, then there had to be a tree for it to grow on, and the molds and things that lack lignin became better able to compete and grow up into the sunlight when they were able to grow tall, and to grow tall you have to be strong, and therefore lignin has a use. And the same kind of reasoning will be expanded when we talk about the plant physiological use and other relationships of these substances. But there is a second corollary then to the fact that these compounds are present in some plants and not in others that I think adds to their importance. In other words, I think they're important because they're secondary compounds. And I'd like to just hope I can clarify that thinking in a simple enough way that you will agree with me without taking a lot of discussion. If a compound is essential for life, then certainly the creature has to produce it. And therefore, it isn't subject to variability to the same degree as if the plant didn't have to produce it. So if you have secondary compounds, they can be environmentally responsive, for instance. Whether or not they're there could be related to the environment. They can be related to pest resistance. So that again, as in the Dalbergens, for instance, the indication is that similar plants don't have them, cannot resist pests as well as those who do have them. Therefore, the fact that they were secondary compounds has some value because the plant is free to experiment, mutate, come up with new compounds, and then if they have a use, they should survive, and that plant should outbreed, populate the plants that lack that characteristic. And some of these things can be pretty apparently minor, but may be crucially important. For instance, and we'll come back to mentioning this later on, blue is a color that bees see very easily and don't see red very easily, whereas birds, on the other hand, see reds easily and don't see blues very well. As a consequence, if you have an Australian bottle brush in your front yard like I do, you must be prepared to be awakened by hummingbirds at 6 o'clock every Sunday morning because the hummingbirds are attracted to the bright red flowers and come to get the nectar and incidentally pollinate the plant. Whereas if, on the other hand, you could breed a blue bottle brush, why, then you wouldn't be troubled, we assume, with the hummingbirds. Anyway, it is true that naturally blue flowers like lupin and so on are bee pollinated, and naturally red flowers, particularly tropical red flowers, are generally bird pollinated, particularly hummingbirds. So that this would be a mutation, clearly, that would give the plant a hummingbird flies a lot further than the average bee, and it would give you an extension of your propagation area to some degree, I suppose, to go to bird pollination. On the other hand, if there aren't any hummingbirds and you have only red flowers, you're certainly at a disadvantage. So this kind of thing, I think, is extremely important, and I think it's important to sort of glory in the fact that phenolic substances are secondary rather than essential for life compounds. A corollary to that, if you like, or at least an argument in this direction is, remember we've shown that the phenolic biosynthesis is at sort of the intersection of a lot of crucial metabolic pathways, and certainly phenylalanine is essential, and tyrosine is essential, and if you're going to divert phenylalanine and tyrosine to making phenolic substances of other kinds, then clearly you're shorting that metabolism. Now, true, the plant has photosynthetic power of making a lot of carboniferous compounds, but even so, many of the alkaloids which are using nitrogen are also phenolic, and plants are generally desperate for nitrogen, so that you'd be diverting nitrogen to secondary products, in this case alkaloids rather than phenols. Carbohydrate metabolism is involved, another key metabolic pathway. Acetate or fat metabolism is involved both through the isoprene path and through the malonate addition to make flavonoids. So that with these things as diversion of important primary compounds, certainly carbohydrates and protein materials are primary, if you're going to divert them to secondary products, then it would seem by and large there ought to be a reason for this diversion. And other arguments can be made. For instance, cyanidin, apparently, if there's a function of anthocyanins in plants, and there are plants that have blue flowers and they're completely cyanidin-bearing, the cornflower being one, and there are others that have red flowers that are cyanidin-bearing, Chrysler Rose being one. And so if you got cyanidin, you can make reds and blues, and you don't need to have delphinidin to make blue, or malvidin for that matter. So why is there any mutation pressure to develop these additional pigments if one compound is perfectly satisfactory? Well, maybe there is no evolutionary pressure. Maybe it's just a random mutation. But on the other hand, it does appear there might be some important utility. Well, in order to save some time talking about the test briefly, I think I'll just skip over a couple of points and then come back next Wednesday. We have studied the occurrence and distribution of phenols in plants in two major systematic ways. One by the genetic study, and this has been largely confined to anthocyanins, so that people will take a white-flowered plant and a red-flowered variety of the same plant, cross them and see what the genetic development of anthocyanin is, you see. So they can do that visually and don't have to have any chemistry to do it. So there have been a lot of genetic studies that have helped us clarify the situation regarding genetic control of phenolic composition. And, well, I better not take time to say anything further about that. But the genetic studies preceded then chemotaxonomic type studies, which would be the other group, where the actual paper chromatogram or other isolated type of phenol patterns, maps, are studied. And then this is related to the taxonomy of the species, genus, or what have you. And on the last page of your outline, you have a sort of a family tree going from the bacteria clear up to the angiosperms, indicating that the lower plants lack flavonoids. And then the mosses begin to have a few flavonoids, and the few flavonoids would include 3-deoxyanthocyanins, so that lack of the 3-OH group, which would mean you couldn't have the usual type of glycoside, a usual 3-glycoside, could not occur. So that the flavonoids are rare, and the ones that are there are rather unusual from what are considered common in the higher plants. And then the continuation on up. Generally speaking, then, since flavonoids are absent, the phenols of the mosses and lower tend to be acetate-derived. You probably noticed that the lignin-type phenols we've been pointing out all appear to have meta-hydroxy substituents, and while it is true that some lower plant phenols are shikimate-derived, apparently the lower plants never learn to hook the two together. So they can't make flavonoids, even if they can make certainly tyrosine by the shikimic pathway and many other phenols by the acetate-type pathway. At the bottom of the page 4, we have a few suggested in higher plants, primitive, advanced, and isolated characters. And there are a couple that I'd like for you to add to your list. One is a typographical error. Under advanced characters gain mutations, that says the OH. It should say tri-OH. So the tri-hydroxy in the B ring, or the pyrogallol type analogs, is a gain mutation that is fairly advanced. So the production of delphinidin is an advanced character. And then at the bottom of the page, you might indicate, not under isolated characters necessarily, but one point that I wanted to make is that the woody plants tend to have condensed tannins, or at least tannins in them, as opposed to the herbaceous. The herbaceous being the grassy type things that are not woody, do generally lack tannins, particularly condensed tannins. And also, interestingly enough, the monocotyledons apparently lack ellagic acid. Also some indication that gymnosperms and ferns lack ellagic acid. So at least monocotyledons and the very lowest type of higher plants seem to lack, and of course the lowest plants of all do too, lack the ellagic acid. So ellagic acid seems to be what might be called a hapactic mutation, and that means one that only occurred once. So somewhere in the development of plants, the production of ellagic acid mutation occurred, and apparently it can be traced back evolutionarily, and it only happened once. And we may say more about that later, but at least I wanted to say that much. On page 3, I'd particularly like for you to read over some generalizations as to what might be fairly typical in the occurrence of phenols under chemotaxonomy. Since my time is essentially up, I'll pass out the test then, and remembering